Mercury News, February 25th, titled "Flu Season's Late and Low Return to the State." Midwinter similarity in 2011 strain cited as reasons for virus's delay. That was the subheading. Thought you were spared. Flu season is hitting California finally. The United States is experiencing its latest flu season in nearly 3 decades, and California is one of 2 states now reporting widespread activity with the typical sore throat, stuffy noses, nagging coughs, and achy muscles. While experts can't say for sure what took the flu bug so long to show up, some suspect milder weather may have helped slow the virus spread. It often has its heyday during the cold winter months when large numbers of people gather indoors. And because this year's strains are so similar to last year's, more Americans may have built up immunity either by getting sick last year or being immunized. This year's vaccine is a good match for the circulating strain. And then the article goes on to talk about past peaks and how February generally is the month in which we see the most flus, I guess, if you will. It's the month that generally serves as the peak. We're seeing that being pushed out now, and so March may be the month that we see the highest flu, or maybe even out to April. Anybody here sick? Anybody here been sick recently? Okay. Anybody plan on getting sick? Anyway, I just thought I would bring that in, let you know that the flu virus is still out there and has finally made its way to California, and hopefully you'll do the right thing to avoid getting sick. One of those things, wash your hands. The more you wash your hands, the better chance you have of avoiding the flu virus. And of course, stay away from sick people. That always works too. So last time we were talking about the structure of the prokaryotic cell. And if you remember, we had a cell membrane surrounded by a cell wall. Outside that cell wall, sometimes there was a capsule called a glycocalyx that could be entire or it could be diffuse. Diffuse just means kind of irregular and broken up. We mentioned that many microbes have a flagellum. Some of them maybe have more than one. The flagellum can be polar, like I've drawn here, or it could be peritrichous, in which they're all over the cell. We mentioned that the genetic material is a singular loop of DNA. It's circular. When you see it drawn in pictures in your book, you'll see it all squiggly crammed into, just to give you the idea that it's a really very large molecule. But it is circular. It is a single chromosome. So there's the genetic material, no nucleus. There's no envelope surrounding it. And then we have in here ribosomes that will be present. You could have storage vacuoles, plasmids, which are extra pieces of genetic material picked up from other microbes. Pili, little structures that extend off of the cell membrane that serve for attachment and movement. One point of clarification with regards to exocytosis: bacteria don't carry out exocytosis. Bacteria don't have any internal membranes, so that process doesn't apply to them. When bacteria need to move products in and out of the cell, they're going to use those protein receptors that we talked about, those protein carriers, or protein channels. That's the only process that they'll use to move things in and out of the cell. You remember we talked about the cell membrane, the structure of the cell membrane. It's a phospholipid bilayer. We talked about movement of molecules across this membrane. Generally, Diffusion is not a process that bacteria employ much, simply because it requires a concentration gradient, and that concentration gradient generally is not established to allow for diffusion. Osmosis can occur as well. Most of the movement across the membrane, though, is going to be active. Some kind of active transport requiring energy, requiring the input of energy to move those molecules into the cell. Generally because you're moving against a concentration gradient. Anytime you're moving against a concentration gradient, you're going to have to put in some energy. We talked about the cell wall. And the cell wall, there are two fundamental types. There's the gram-positive and the gram-negative. The terms tell you about how they accept stain, but it also tells you much more than that. You know, a gram-positive microbe, then, is going to be a microbe that's going to have a very thick peptoglycan layer. And that a gram-negative microbe, then, is going to be one with that thin peptoglycan layer, but it's going to have that outer membrane. And that's significant when we describe the bacteria, because it really plays a role in what can affect them. So a gram-positive microbe, cell membrane, lots and lots and lots of rows of peptoglycan. Gram-negative microbe, inner cell membrane, one or a few peptoglycan layers, and then an outer cell membrane. And remember, this outer cell membrane contains a molecule that's uniquely bacterial. The lipopolysaccharide leaflet of that outer membrane is unlike any membrane in any other organism. Bacteria are the only ones that have those lipopolysaccharide molecules. And we talked about the two parts of the lipopolysaccharide. There was the O-specific molecule. Remember, that kind of served as an identifier, helped us identify the different types of cells. And then there was the lipid A molecule, and that was the one that served as the endotoxin. What that means then is that part of the microbe itself plays a role as an endotoxin, triggers an immune response. Now, if the exposure to the microbe is small, your immune response might be small, minimal, and you defeat the microbe. But if the microbe is in the bloodstream and multiplying, then the amount of exotoxin present increases, which would increase the immune response, which could cause damage to your own cells if the immune response was too great. Any questions? So this is like I said, this is a fair essay question. Have you diagram the structure of a prokaryotic cell and identify the parts. This also is a fair essay question, having you compare and contrast gram-positive, gram-negative cell walls. And we also talked about run and tumble, the way the bacteria move. This too now is a fair essay question. I tell you this because you should answer each one of these questions to yourself. Maybe sit down and write it out even before you get to the exam, so that when you see it on the exam, it's not a surprise and you've already written out the answer to it. I don't know why these are always so wet. Let's turn our attention now briefly to the eukaryotic cell. I got the override. Well, first thing that we're going to note is that the eukaryotic cell is substantially larger than bacterial cells. Generally on the fold of about 100 times larger. Now, of course, there are exceptions to that, variations to that. We have found